Yes, Janvi. Good evening. Am I audible? Jaishnavi. Am I audible? Yeah, I'm not audible. You're not audible, sorry. I can't hear you. Yes, Jaishnavi. Can you hear me? You are not. So let us start. 5 minutes. I don't know when the next one. So we are going to. Can you hear me, Jaishnavi? So we are going to study about cells and tissues, right? Study of cells, cytology. Very good. And study of tissues, histology. Very good. So I will show you. And I will say that concentrate on, focus on diagrams also. Because diagrammatic questions comes in the achiever section. This will take this also. Okay, so Robert Hume, he discovered cell, right? In the cork cells. Cork cells, you know, it is the bark of the outer part of the tree. That is ruptured fat. That is dead cells. And he observed under microscope, right? And that microscope was observed, discovered by Anton van Leeuwenhoek. Slide show. We can remove this one. No, more. Yes. There is an option. Right. We do panel. Status. There. Start from the beginning of the video. Hey. This one. There is another one. This floating. Yes. This one. Sure. Sorry again. What did you want to hide? I want to hide this one. Oh, you want to. There is a. But now what should I do? There was the slideshow, yeah. Okay. But I can't see you now. See her, I can't see her. The TV is not good actually. You want to see her. Let it be like this. Jaishnavi, can you see this PPT? Is it visible? What is that? मुझे पता ना if she is there or not. ये वो पैनल हटाना तो मुझे वो स्क्रीन ही चला गया स्टूडेंट की नजर है. क्या मीटिंग से ऑफ हो गई क्या? निकल गई क्या वो? हाँ. स्टूडेंट अंदर नहीं है. शो मीटिंग में है क्या? शो मीटिंग में है. मोड पे जाके कुछ किया था. ये मोड पे देखो. Jaishnavi, is the PPT visible to you? स्टूडेंट नजर नहीं आ रहा. चलिए ऐसे करा. नहीं स्टूडेंट को प्रॉब्लम वो दिख रही है वो दिख रही है ना अंदर है. ठीक. एक बार जी आवाज से. Yes, Jaishnavi, she is giving the response. Jaishnavi, are you there? Response. Jaishnavi, can you hear me? Am I audible? अच्छा. पर उसकी आवाज मुझे क्यों नहीं आ रही? If she wants to ask any question, कैसे पूछेगी? Unmute yourself, Jaishnavi. I can't hear you. इनका प्रॉब्लम है क्या? आपका प्रॉब्लम जा रहा है. ठीक है, करवा दे. ठीक है, किसे इसको करवा दे. Actually, नहीं ठीक है ऐसे करा. यार है ना नजर ये हटाना. चलिए ऐसे करा. हाँ. अच्छा बस वही करना था क्या? So we were discussing about cell, right? So there were two scientists, Schledel and Sean. Huh? They just he is a botanist. Schledel and Sean is a physiologist. He studied on plants. That is, he's a person who studied plants. And he composed of the cell. He told that plants are composed of cells. The animals are also composed of cells. So Theodore Schwann also just took the outer layer of the plasma membrane, found that plants also have cell wall, right? So they told that both plants and animals are composed of cells. Now later on, Varsho, he proposed that cells are composed of they exist of pre-existing cell only. Like if the cell was already existing in the environment, it just there is a separate division and it divides into two like this. First he just elongates, and like this it elongates. The cell elongates, nucleus elongates. Then there is a division like this in the cytoplasm. It's divided into two like this, and then divided into two separate offsprings. This we'll discuss. Okay. Means this cell has already coming from the existing cell. So Varsho explained that cell arises from the pre-existing cell. So these 3 points were considered as a cell theory. Next, all cells have ribosomes and cytoplasm. Cell organelles are present in the cytoplasm only, and ribosomes are present in. You must have studied about the chloroplast. Ribosome is present there, and see here, this endoplasmic reticulum, ribosomes are present over there, and if endoplasmic reticulum, if there is a ribosome, then it is RER, pilotenement, and mitochondria. And then there is a different size and shapes of cells. Cells are of different size and different shapes. Right? Then see here, the smallest cell is bacteria in PPLO. PPLO means Neuromonia-like organism. Okay, it is from 0.1 to 0.3 micrometer. And this is the type of bacteria only. And virus is smaller than bacteria, but we do not consider virus as a cell. You know why? Yes. They don't have cell membrane. They are just made up of proteins, protein structure and all. So that is why we say the smallest cell is bacteria, and the longest cell is neuron, nerve cell. This is ostrich cell, right? And these are bacteria, and this is RBC. And based on the functions, cells have different shapes. These are plant cells, and these are animal cells. Okay, they may be tubular, oval, or circular, cuboidal. Okay, and according to the shape, there are two types for your prokaryotic cells and the eukaryotic cells. In prokaryotic cells, they consider mainly bacteria and blue-green algae. Blue-green algae are cyanobacteria. Cyanobacteria. Remember these terms. They may ask blue-green algae, cyanobacteria. So, see, bacteria have different shapes. This is rod shape. This is circular shape, right? There's spirillum vibro shape. See, this is rod-like. This is spherical. If it is one, it is monococcus. When it is two, it is diplococcus, staphylococcus, right? When it is in chain, it is coma-like and the spiral. This has a shape of bacteria. And if you study the bacteria, we see that the outermost layer is capsule, and it has 3 layers. And inside this, there is a cytoplasm. Now, in the cell, you can see this is DNA. But if you remember eukaryotic cell, animal cell, or plant cell, it is circular, means it is membrane bound. So nuclear membrane is not well developed in prokaryotic cell. This is the basic difference between eukaryotic cell and prokaryotic cell. These are lying like that, and these are ribosomes. It is present here only, and these are plasmids, and plasma membrane cell wall is the middle layer. If we see here, these 3 layers are there. The outermost part is glycocalyx, the central part is cell wall, and the plasma membrane. Tightly bound layers are there. Glycocalyx is the outermost layer, maybe slightly or capsule shape, right? And plasma membrane, there is a folding in the plasma membrane. This is a plasma membrane. No? There is a folding like this. And this folding is called mesosomes. Okay, and mesosomes have various function like cell wall formation, application, DNA application, and chromosome to total cell distribution, respiration, integration, increasing the surface area. And mesosomes have various functions. And these chromatophores are present in cyanobacteria. These are pigment foldings. So mesosomes and chromatophores are the membranous structure. And nuclei, as we have discussed, these are DNA, and outside there is another that is called plasmids. This plasmid is a circular DNA. Okay, outside this DNA, and they have a phenotypic characters. Plasmids. They give resistance towards the bacteria. Okay, that is antibiotics. Immunity, yes, basically they provide immunity because they have a resistance to antibiotics, right? Then flagellum is the locomotory organ. So it is divided into three. This is filament. Filament is the larger part, major part in this. Then middle part is hook, right? And this is basal body by message, right? And along with this flagella, there is this pillar. These pillars, fimbriae, these are also called fimbriae, elongated tubular structure. Okay, they just attach to the bacteria, to the rocks, to the streams, okay, and or to the host tissues. So this fimbriae, their internal structure, and ribosomes. As we have discussed, we have told that these are freely present in almost all the cells, right? In bacteria also it is freely present. See here it is freely present. Now bacteria, ribosomes are site of protein synthesis. Means protein synthesis, proteins are synthesized in ribosomes. Whether it is freely available or it is attached with this very endoplasmic reticulum. And these ribosomes, they have two subunits, 70S and 80S. 70S is in the prokaryotic ribosome and 80S is the eukaryotic ribosome. It is present in the eukaryotic cells. Now, this S is the Swedberg unit. It tells the sedimentation coefficient. Means, see, when they just, there is a centrifugation, when the cover, that particles settles down, that is sediment. So that is sedimentation. They just find out that sedimentation coefficient. Okay. And this 50S and 30S are the subunits. This is the 50S is the largest subunit and 30 is the smaller subunit of 70S. And this is not the adding up of this. This is 50 plus 30 is 80, but it is 70S. No? So it is not the simple addition. It is like how well it settles down. Okay, right. Then this is the way how it synthesizes proteins. Means the larger subunits and smaller subunits, they just attach to this strand. This strand is mRNA, messenger RNA. Okay, and these purple one that is shown here, it is polypeptide chain. Polypeptide chain is the amino acids, chain of amino acids. And it is also called the polyribosomes. Now, inclusion bodies are non-cellular, non-living cells, no? They may be granules, gas vacuoles, they may be, okay, or see here, they're lying here, cytoplasm only. They just non-membranous and they just preserve material. It stores materials. Prokaryotic cell you have understood? Just just remember the figures. Okay. Otherwise, it's studying this the next time. Okay. These are eukaryotic cells. You can see here basic differences in nucleus. This is membrane bound. Second difference is that you will find that there is a cell organelles in the embedded in the cytoplasm. Whereas in prokaryotic cell it wasn't there. Right? Cell membranes were not there. All the cell membranes are absent except ribosomes. Ribosomes are present. See here, this is a plant cell and this is animal cell. The basic difference between plant cell and animal cell are eukaryotic cells. The basic difference between the two is very large vacuoles. See here it is large vacuoles. They have plastids in the cell wall. And endomembrane systems are endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. In the 9th standard, we have studied that all these are cell organelles. Yes, these are all cell organelles, but these 5 are called endomembrane systems. Why? These are called endomembrane systems because they are linked together with each other. See, let me show. Here they go. This is endoplasmic reticulum. It is attached with the Golgi apparatus. And the lysosome is just formed in endoplasmic reticulum only. And this ribosome synthesis is there in this endoplasmic reticulum. Sorry, protein synthesis is there. And these proteins enters from this to the trans surface, and the lysosomes also come out, vesicles come out, right? This I will discuss. So this is a plant cell. I think basic structure you must be knowing regarding this. For this, now this is cell wall. Cell wall is made up of cellulose, pectin, galactose, and mannans. And let us study the endomembrane system first. These 4 are endomembrane system. This is endoplasmic reticulum. You see, this is nucleus, clear membrane. This ER is attached with this, and there is no ribosome means it is. There is no ribosome means it is smooth ER, and this is rough ER. The main function of rough ER is protein synthesis because ribosome is present. So ribosome is not present here. Then what is the function of smooth lipid formation? And function is lipid formation. Fat cells or cells. This is Golgi apparatus. It has 2 surfaces, convex and the concave. This is this surface and this is trans. See, the protein that synthesizes in endoplasmic reticulum, it just enters from this phase. It stores here, they converts into starch or phospholipids, and then it just comes out from here, secreted in the form of secretory vesicles. Okay. These are newly, this round form are the newly forming vesicles. These are cisternae. These are called cisternae. Okay. Different there. Usually we just mix cisternae and cristae. Cristae is present in mitochondria, and this is cisternae. So function of Golgi apparatus, lysosome formation, right? Apart from that is glycoproteins, glycolipids, and these are lysosomes. These are considered as the acidicidal. That's why. Because they contain enzymes which help digesting. Yes. Okay, lysosomes are found in Golgi bodies. Right? Very good. So next is vacuole. Vacuole, this is a central vacuole plant cell. There is very small or no vacuole presence in animal cell, right? They contain water, sap, excretory products. Okay, and the outermost outermost layer. It is bounded by outermost layer single membrane, and that is chloroplast. It was there in 9th standard, chloroplast. Right? It actually covers near about 90% of the volume of cell. Right? Then see here, in plants, it is in the center. In amoeba, it is present as in the form of contractile vacuole. Okay, and that helps in the excretion. And in paramecium, it is just like a in the form of food vacuole. Food vacuole means it engulfs, digests food. Paramecium, engulfing. See, this is the way the endomembrane system. See here, ribosome formation is there. Ribosome is making synthesizing protein. From here, it enters into Golgi bodies. Then this is lysosome formation. It just comes out from the trans surface, right? Or vesicles, secretion vesicles comes out from here, or it just goes out of the cell. That is why these are called endomembrane system. See, vacuoles, lysosomes, Golgi bodies, endoplasmic reticulum. All these are related to each other. But what about mitochondria? Why we are not considering mitochondria in this? Mitochondria is a cell organ. Why we are not considering? Because it is not related. It is the energy producing. Yes. Apart from that, it has its own DNA ribosome. Doesn't require the help. The one who don't require any help will always remain. Right. This is mitochondria. See, this is the outer membrane. This is inner membrane. This green part is the matrix, or this is inner membrane, and here it comes cristae. Okay, the main purpose of this cristae is to increase the surface area. Wherever there is an infolding now, it increases the surface area. Like, okay. Yeah. Okay, the diameter is 0.2 to 1 micrometer, and the length is 1 to 4.5. Okay. This is the internal structure. Here you can see ribosomes are present freely. And this is outer membrane. This ribosomes are present in matrix. And this is DNA. These are the ribosomes. It is present and helps in the protein synthesis. The chain of amino acids. You know? And has an intermembrane space here. These are cristae. And the main function of mitochondria, as we have told, it helps in it. So, how this energy is here? Because ATP synthetase. Because of the ATP synthetase enzyme that helps in the formation of energy conversion cells, that is ATP. These are. Next, plastids. Plastids are present in plant cells only. There are 3 types: chloroplast, chromoplast, leucoplast. Chloroplast means it contains chlorophyll. Chromoplast, it gives color pigment. So it is the color pigment. It gives colors to the plant, and leucoplast, starch. It is colorless, like in potato. Okay. Now the chloroplast, these are present in the mesophyll cells. This is stroma. This is a stack. It's called thylakoid, stack of grana. Okay. And this enclosed space called lumen. See here, these are lumen present in the thylakoid. This is stroma, and photosynthesis takes place in this chloroplast. Yeah. In 10th standard, you have studied light reaction and dark reaction. Light reaction, it occurs. Which part? Stroma or grana? dark reactions and stroma light reaction. Okay. So these stroma and grain are held in photosynthesis. Now ribosomes was first observed by George Patton. Very important. Complete name of ribonucleic acid RNA and the proteins. These are non-membranous spheral structure composed of these two. It has two subunits we have discussed, right? Now next is the centrosome and centrioles. These centrosomes and centrioles are present only in the animal cell. It's not present in plant cell. Okay, and the main function of this centriole and centriole is spindle formation in mitosis. Mitosis and meiosis is there in your syllabus. Yes. So this spindle, yes, correct. Spindle formation is because of this centrioles in animal cells. And what about plant cells? In plant cells, we were not taught about this. We were just told that the cytokinesis occurs in this. Okay. Let me show. See, microtubules. These microtubules are present here. These microtubules help in cell formation, and that helps in cell division. Okay, these are microtubules. Okay. Now the centrioles are stack of 9. This 9 evenly spaced peripheral fibrils, that is tubulin, and that is in the triplet. Okay. It helps in this central is present in centrosome and it helps in spindle formation. That lie perpendicular to each other like this. And see here. Perpendicular to each other. Next is nucleus. What do you know about nucleus? Nucleus is like the brain of this that you can see on the and it contains a structure of nucleus. It contains a nuclear membrane and inside is the nucleoid. And this is not nucleoid. Nucleoid is in prokaryotes. Nucleolus. Yes. And the nucleolus contains the chromatin. Nice. Yeah, this is nucleolus. Outside this is here chromatins are present. It is not inside that. Inside is the DNA formation. RNA formation. This chromatin contains this long thread-like structure chromosomes. See here, this is nucleolus, the chromatin, and this VR and all, and nuclear alveolar. Okay, or nuclear membrane, and it has pores. Okay, so interphase nucleus contains these four. Nucleoplasm means instead it is cytoplasm in nucleus, it will be nucleoplasm. Matrix it is. Remember like this, Now this nuclear membrane, it has pores. So if it has pores, the what will be the function of this? For exchanges. In this case, and inside it is RNA, so there will be the movement of RNA and protein. And cells means solute ions, but in nucleus it will be RNA and proteins. Now chromatin, it is a network fibers, and that is made up of DNA and proteins, right? And that is why there is the exchange of these two, and these forms chromosomes. This is chromosome. Nucleolus, we have just told that there is RNA, so no need to discuss. Now this is chromosomes, chromatids. These were sister chromatids. So this is the center one is centromere. It is also called kinetochore. Okay. This is chromosomes are like this. And then maybe how many chromosomes are there? And if we talk about it pairs, 20. See, on the based on the type of this position of centromere, It is of 4 types, right? If it is in the center, it is metacentric. If it is in the little above the center, it is submetacentric. Acrocentric is towards the close to the end, but in telocentric it is at the end. This is telocentric. This is acrocentric. This is submetacentric, and this is. And sometimes there occurs the secondary part that is called satellite. Secondary construction. Okay. Now let us. Which of the following statement is incorrect? Cytoplasm is also known as protoplasm. Lysosomes are known as inside the bag of the cell. Mitochondria has its own DNA. All of the above are incorrect. None of the above. Incorrect. Read it again. Cytoplasm is also known as protoplasm, where it is together with nucleus. And nucleus and cytoplasm together forms protoplasm. Rest of the statements are correct. See, क्या होता है ना, in such questions, the answers are very nearby, which we think that it will be correct, but sometimes that's not correct. Huh? Second, which of the following is not a function of vacuole in plants? Toxic metabolic waste. They help in the process of cell division. They help to maintain turgidity. They provide structural support. None of the above. Very good. Where are the essential proteins and lipids required for cell membrane manufactured? Lysosomes, chromosomes, endoplasmic reticulum, mitochondria, none of the above. Endoplasm. Okay. That is SCR. The process by which water moves through a semi-permeable membrane from a region of high concentration to a region of lower concentration. This you have studied, isotonic solution and hypertonic and hypertonic. These solutions are there. Hypertonic, hypertonic and isotonic. What is there in hypertonic solution? When we take a cell in hypertonic solution, exosmosis. If exosmosis occurs, means there will be the shrinkage. You might have observed that while leaving your fingertips, working in the water, your fingertips, there is a shrinkage in the fingertips. That is because of hypertonic. Exosmosis occurs in this. And you must have seen current. Current means kishmish that if you put it in the water, then what will happen? That water will enter into that. So why will enter? Because it is placed in a hypertonic solution, osmosis occurs, and there is no there's a net movement of water in acetonic, equally movement. So the osmosis are of two types: exosmosis and endosmosis. When water enters into the cell, that is endosmosis, and when it exits, exosmosis. Okay. Now, we have discussed about mitosis and meiosis, that central cells in the spindle formation in mitosis in animal cells. So, there occurs the cell cycle. Means that is the life period of the cell during a cell that synthesizes DNA, grows, or it divides into two daughter cells. See DNA, how it is replicating. This is called DNA replication. When the two strands separate and each strand develops the another one. That is the DNA replication. And see here, one cell is replicating and it divides into two daughter cells. Right? So this synthesis occurs in the cell cycle. So cell cycle has basically two phases. Interphase, this is interphase, and this is M phase. Mitotic phase. This is mitotic phase. This is about 90 to 95% it covers interphase. This is M phase. And it is in between the two successive M phases. Next is this interface. It has three phases, two gap phases and one synthetic phase. This you have studied 10th standard. G1 phase, synthesis phase, and then G2 phase, that is gap phase. G1 phase, what occurs? The organisms which have their own DNA. There's a growth. For the first gap phase, there's a growth. Cells are metabolically active. DNA replication is there. Synthesis of RNA and proteins all occurs in this gap phase. Okay, and then the synthesis phase, the DNA gets doubled. The amount of DNA gets doubled. See, there is a 2, now it is 4. It happens in synthesis phase. But what about chromosome numbers? It will remain same. The chromosome numbers of all animals have different number of chromosomes. Like chromosome, like human beings, they have 46. And so it will remain the same, but there will be the replication, double of this in mitosis. And here this replication begins in nucleus because I've shown that it has DNA, the replication occurs in that. Next is the second gap phase. Same, it continues, cell is prepared for mitotic phase. But sometimes some cells cannot go further. Then what happens? That they enter to the rest. That is gap 4, and this stage is called quiescent stage. And then they start the cycle again. And M phase, mitosis phase, means there are two kerokinesis and cytokinesis. Kero means there is a division in nucleus, and cytoplasm here. You can see here there is a division in these cells. Right? So this is a kerokinesis. But in this phase, see, when they enter into telophase. This you have studied in the telophase. Then what happens? That there is a separation in division in cytoplasm, not here in the center. That is the endocytokinesis. Yeah. Now this is in mitosis. Various phases. First, it is the interphase. When the cell division starts, it just interphase. It just forms chromosomes. It's chromatin material. The chromatin material. It long thread-like structure. It just converts into chromosomes, centrioles, move parts, and these centrioles move apart and move towards the poles, and they start forming spindle formation. And in metaphase, then this in metaphase, these chromosomes lie on the epidermis. Yes, and in anaphase, they lie into poles. And then afterwards in telophase, there is a division in this. What is this called? Furrow formation. Furrow formation occurs in this. Actually, it starts in anaphase, and in telophase it gets develops, and then it just divides into two daughter cells. Here they are dividing into mitosis is dividing into two daughter cells, but the chromosome number remains the same. And then meiosis, see here, this is a cell with a pair. It gets duplicates and they form tetrad formation. Here there is the replication. Okay. Then what happens? These two chromatids they just to crossing over, and then they get separate. See here, this like this, then this portion and this portion. It will come from here. There is no. So what will happen? Suppose this part of this chromosome. Yes, it will come this side and this part will come this side. See here, green portion. It is not clear in diagram. Green portion here This yellow portion plus green portion will come here. This green portion with the yellow part will come here. So after this, they get separated out. So in meiosis, there is a formation of 4 daughter cells, whereas in mitosis, there is a formation of 2 daughter cells. Diploid cells. And these are haploid. They are single, no? So they are considered as haploid. And here they are in the 2, so diploid. Is it clear? Okay. Now DNA replication is controlled at which of the following checkpoint? G1, G, M or S? Starts G1. Then cells will no longer divide, such as certain specialized cells found in the human brain remain in which phase of the cell cycle? Phase S, GO or G1? GO. A centromere is another name of kinetochore, a structure of which microtubules attach, a constriction on a chromosome bound to a disc, all of the above. All of them. Structure with a microtubule attach, centromere. Sorry, answer bata diya. The current sequence of phases of the cell cycle is? You just recall cell cycle and tell me the same. GO is the last stage when this enter into the rest of these. Then you can start on G1. G1, S, G2, T. Okay, GO has the GO is. If there will be GO, then next this up to cell it is clear. Everything is clear. Now next is see all cells perform some specific functions, right? Plant cell if you consider, but they have different cells. Animal cells they have different cells. Muscle cells have different cells. What is the function of muscle cells? Movement. Okay. Skeletal tissues. Movement. Muscle tissue and skeletal tissues work together. Okay, even alimentary canal, they have different functions. They have different organs. Each organ has different tissues. Tissues are composed of different cells, right? So they perform different functions. Let us start with the plant tissues. Why have made this mind map flow chart? Because sometimes they may ask sclerenchyma is the part of which type of tissue. They can ask complex or simple, or they can ask myostatic or permanent. If we know this chart now, we simply write that sclerenchyma belongs to permanent. So plant tissues are of 2 types, meristematic and permanent. Meristematic again 3 types, and permanent are 2 types. This permanent again are divided into these 4 and complex again into these 2. We'll discuss each of these one by one. See, let us discuss from the meristematic tissue. These are the actively dividing cells. Meristematic tissues are actively dividing cells. It is usually present in the tip of the plant. If it is present in the tip of the plant, that is apical. If it is present in the nodes, okay, lateral, and if it is present in this with the nodes, it's see here. This is the cell, meristematic cell. Cell nucleus is present in itself. It is very thin, and main function of this meristematic tissue is they lack vacuole. You know why they lack? Sorry, main function is that it divides. And but these here you can see in this cells they don't have vacuoles. You know why they don't have vacuoles? Yes, they are actively dividing cells. If they are actively dividing cells, then they are getting the nutrients. So there is no need of storage. So they don't require vacuoles here. and it has in the decrease of that and girth, right? These are three. These are present on the tip, it is apical tip, and if it is present at the internode, it is intercalary, and if it is present on the side of the root, see here, side of the root, it is lateral meristem. Internodes, intercalary, interyanus. Then see here, apical, it has primordia and lateral bird primordia. Now, this you have understood the three. Then simple permanent tissues. Actually, the permanent tissues arise from or dwells from mesomeric tissues. When these mesomeric tissues stops dividing, they by the process of differentiation, they convert into permanent tissues. And permanent tissues are three types: parenchyma, collenchyma, and sclerenchyma. So this is parenchyma. See the cells. It has primary cell wall, nucleus here, vacuole, because it is a plant cell. Now, in meristematic tissue, just now we have started that there is no vacuole, but in permanent tissue, vacuole builds. Why? Because these are not actively dividing cells, right? And if these are not actively dividing cells, means they require nutrients, stored food. So vacuole, main function of this vacuole is storage. So parenchyma cells helps in provide storage. Okay, provide support to the plant, and see here it has intercellular spaces in between, and these are found in the soft parts of the plant. Next is if it is in contains chlorophyll, it helps in photosynthesis. And that type of parenchyma is called chlorenchyma. And in the plant running aquatic plants, they just float on the surface, right? So there must be the exchange of gases. So these parenchyma, because they have air space, na? So they just have in the buoyancy and they float on the water. So that type of is called parenchyma, is called parenchyma. Next is collenchyma. See, these are elongated. It has chloroplast in this. Nucleus is there. Long vacuole is there. There's a intercellular space is there. Okay, and but it is lesser than parenchyma, collenchyma. Okay, and this collenchyma provides flexibility. You must have seen that the young branches, if you try to pull this, it just gets bent. So because of this collenchyma, it is present in the leaf stalks of the epidermis also, and it allows easy bending without breakage. Then sclerenchyma, this has a lignified thick wall. Lignified means it has lignin in it. This lignin, keep it in mind. There is no intercellular spaces, but they have a simple pit. See, they have a simple pit. And this is long, narrow, thick wall. These are dead cells. And it provides strength and rigidity to the cells. Have you seen the outer shell of the heart or coconut fibers? These are sclerenchyma. Nuts and husk of the coconut. These are sclerenchymatous tissues. And if you just see the lower side of the leaf, if you see the veins, that is hard portion. Center midrib, that is sclerenchyma. So it makes it hard still. Because these are dead cells. Next is the cuticle, epidermis. Epidermis is this upper layer, and it has cuticle. Cuticle is just the upper layer, waxy layer on both sides of the leaf, above the epidermis. So function of this cuticle is yes. or it provides just prevents the related pathogens. And below this epidermis, this is peristate mesophyll. These are veins. These are spongy mesophyll, and this epiphyll. These are intercellular cells. And this is stomata. These two are guard cells, and these guard cells present in the lower epidermis helps in or regulates in guard cells regulate in exchange of gases. Very good. This is a cork cell. We have discussed this in the starting. This is the bark, outermost part here. Okay, and it consists of cambium. Below this bark, there is the cambium, and this is sapwood. This is hardwood. This is little darker, and this is lighter, and it is indicated. All the nutrients flows to the element in this part only, in sapwood. And this hardwood has growth rings. Growth rings means it tells the age of the tree. And this bark is the ruptured epidermis is that is bark, right? It is called thick, and there is no intercellular. And these are epidermis, cork cell, these all are protective cells, tissues. Now, stomata, as we have discussed, these are guard cells. Kidney-shaped guard cells are there, and these are showing closed stomata or open stomata. So these are regulated by guard cells, right? And these are the epidermis cells. And where the stomata is present, and the leaf, the name and the side. Next is the complex tissues. Complex tissues are permanent tissues. Types xylem and phloem. And these are also called vascular. Yes, vascular bundles. Now, if we study about xylem, we see here, we find that it consists of tracheates, fibers, xylem fibers, xylem parenchyma, xylem vessels. Xylem parenchyma is there, it will help in storage of the food. Okay, and these are thick cell walls. Xylem fibers, it provides support. Okay, and because these are present, parenchyma present on the side, so side-wise conduction will be there in this. Tracheates and vessels are tubular, and they just start at ventricles. Tracheates and vessels. So support is given by xylem fibers. It has its storage, and conduction is because of this tracheates and vessels. See here, this red arrow shows the movement of water, xylem vessel, and with water in the soil, it goes upwards, so it has only unidirectional flow, right? These are tracheates. So, but why unidirectional flow? Because the water is not able to go to the yes. Okay, so there is no end wall in between the cells. And this is thick, and it has lignin in it. Pectin is present in pectin. Cell wall, yes. And now this is phloem. It has various elements: the plate, the tube, phloem parenchyma, companion cell, and the tongue. This phloem fibers. These phloem fibers here here I have not shown fluid fiber is been along with this tubular structure, and that fluid fibers are spiral pellets fibers. Okay, and see here, companion cells are present here. What is the function of this? See, cyanoparenchyma is living or dead? Cyanoparenchyma. All cells are dead here. No, rest. And in fluid, including fluid fiber is dead, rest are dead. Cyanoparenchyma is there and fluid fibers are dead, rest are living. Okay, on companion cells, what's storage of the cells, right? Seed tubes have perforated walls here. And a seed is a seed tube. It has perforated walls through which this food enters, right? The xylem tracheids and vessels has in transportation of water. Here in seed tubes only, or in a xylem only seed tubes helps in transportation. Okay, and then they just provide. And this see here how these are attached. This is xylem. Then cambium. Cambium is the and fluid. Right? These in between the two. See there. This is cambium. This is phloem. This red part and the brown part is xylem. Patient in the periphery is like this. Regarding this plant tissues, you have understood? No doubt. Next is animal tissues. Tissues means what? So animal tissues are of 4 types: epithelial tissues, connective tissue, muscular tissue, and neural tissues. Here these are located like this. See, epithelial tissues are present here on the surface of the body. Then muscle tissues here. Then nervous tissues, these nerves. And connective tissues means whose connective tissues are here, and bones, and then cartilage between the joint and nose, tape, and ear, and the blood. So these are animal tissues. So let us take each one of these. Epithelial tissues. Here you see this. It has a free surface, and it is covered. It may be in a single layer or in a multiple layers, and it has a very little intercellular matrix, no space in between. Yes, simple single layer, that is simple epithelial tissue, and the compound having multi-layer. Simple epithelium again are of different types: squamous epithelium, cuboidal epithelium, and columnar. Simple tassels and cuboidal, cube, and columnar is column. Each one of them has nucleus. This red part now, purple and they are pink. These are nucleus. All the nucleus are present in the cells. Simple are 3 types. Now you can see here alveoli. You must have studied alveoli. What is alveoli? Alveoli are the smallest. It's shaped. Very good. So it contains simple epithelial cells. Okay. All the dentate is present in the blood vessels, lung alveoli. So main function is diffusion boundary of the diffusion form. Function of alveoli is this only, no? Stereocapsules. So they have planning cells, and the boundary is just regular. Next is cuboidal. Tube-like cells are there having nucleus in this. It is present in the glomerulus, the tubular part of nephrons or ducts of plants. It is present, and the main function of this is What is the function of, you are doing experimental system? What is the function of this? Everyone? It's the smallest. Means there is some reabsorption and secretion of various ions, salts, and waters. So this cuboidal epithelium main function is secretion and absorption. It is present in PCT, proximal fiber, formulated tubule, and it has microvilli also. This. Next is columnar epithelium. See here, these are tall column-like structures having nucleus located at the base. These surfaces may have microvilli, it is present in the lining of stomach and intestine. So, function will be secretion and absorption for this. Because they are present in intestine and stomach, we know the function of intestine is absorption, and the absorption is because of the muscles and this simple epithelium. Now, columnar epithelium, cilia is present in this column cell. Columnar cell. It is a part of columnar only, maybe cuboidal also. Cilia. So what is the function of cilia here? To push the particles. Sometimes if we cough, mucus comes out. That is because of this cilia is working in our respiratory tract. It pushes the extra particles out, right? Like here, here. And even in this fallopian tube, it has cilia, so that embryo can that cells can enter into that. Zygote can enter into this. So the main function is of this is push the particle. Yes, mucus out. Okay, and it is present in bronchioles and fallopian tubes. Yeah, it's not clear now. Now next is this glandular epithelium. We have studied about the glands earlier also. Whenever there is a in mesosome side, if there is a glandular structure, plus increases the surface area, and it may be two types, unicellular or multicellular. Cellular goblet cells, alimentary canals, and multicellular means like the salivary glands. So intelligent secretion. These are goblet cells. Okay. Extended part of the membrane. Next, let us come to connective tissue. Connective tissue. What do you mean by connective tissue? A tissue which connects parts. Okay, so blood is what? How will you define that? Blood is a specialized connective tissue which consists of platelets and fibrils. WBC. Then why it is considered as a conductor? Because it connects one part of the body to the bit and transports gas. And it is all body part. So this is most abundant because blood is means okay, and it helps in supports and leakage and all connective tissue except that have fibroblasts. Okay, like this, these are fibroblasts. Except blood, all connective tissues have this fibroblast. Means they secrete fibrous proteins. What are fibrous proteins? Collagen and elastin. And they give strength, elasticity, and flexibility to the tissue. And the matrix is polysaccharide. Now, connective tissues, let us study the division. It is of types. Loose connective tissue, dense connective tissue, and specialized connective tissue. This is just now you have heard enough. Loose connective tissues are of two types, areolar tissues and adipose tissues. See, this is areolar tissue and this is adipose tissue. And dense connective tissue, dense regular and dense irregular. And this is cartilage, bones, and blood. Loose connective tissues are of two types, areolar connective tissue and adipose tissues, and they have obviously, except blood, I told that they have fibroblasts, macrophages, and mast cells, and these are arranged in the semi-fluid membranes. See, this areolar, these are beneath the cells. They have, these are muscles, right? These are fibroblasts. This one, fibroblast. They have collagen. The fibroblasts, they have collagen fibers. These are macrophages. These are parts of the areola. This acts serves as a support framework for epithelium. Areola. Next is adipose tissue. Here, these cells are called adipocytes, and fat storage area is this. It has nucleus on the side, and in between this there is a membrane, plasma membrane. And these are seen beneath the skin, just below the skin. What is the main function of this adipocytes, adipose tissues? They just provide cushioning between the internal organs and the skin. And why? Because it stores fats. Next is dense connective tissue. Dense regular means there are two types, tendons and ligaments. This is tendon. Okay. Tendon means it just connects muscles to bones. And see here it is present. And ligament is between the two bones. Ankle bone is showing, ligament is this. Between this is bone, ankle bone. So this is join the two bones, that is ligament and more bone to muscle means tendons. Next is gins, irregular. It has collagen fibers, fibroblasts, very common all, and it is present in the skin and the irregular pattern. Then let us come to the specialized connective tissue. Specialized connective tissues are specialized connective tissues, blood, muscular tissue. Muscular tissue is specialized connective tissue. Specialized, blood, just now we told. Apart from blood, cartilage, and what is this? Bone. Bone is the hardest one, and the cartilage is. These are cartilage cells. Cartilage is in between the bones. So it helps, it works as a greasing. Right? Apart from this, it is, because it is very flexible, so it is present in the nose tip, in the ear, cartilage there. Even if all children they have cartilage which converts into bones in adults. Few cartilages are like this. But the bones of pens are very soft. This is bone. Cells of cartilage is called chondrocytes. And cells of bones are called osteocytes. And cells of adipose tissue, adipocyte. Adipose with adipocytes. This is a bone from the skeleton framework of human body. Just think if skeleton will not be there, how we able to look like that? To the framework, right? It has the cells are called osteocytes. These are arranged like this of backbone tissues. These are hard and they have calcium salts and collagen fibers in this. These calcium salts and collagen fibers give strength to the bone. The main function is structural framework. Apart from this, it helps support and protect the softer tissues and organs and limb bones in various functions and locomotion and movement and the blood cells are produced. See, this is a long bone. In this long bone, there is a production of red blood cells. So these are called this is called bone marrow cavity, and there is a production of red blood cells. So the blood cells are formed in this bone marrow. You will say bone marrow. So let us start with this blood. What is the shape? Different components of blood. Erythrocytes, and erythrocytes are also known as. What is the shape of RBCs? And what about this WBCs? Amoeboid shape. Okay, and platelets? Irregular shape. And these are considered as fluid connective tissue. Okay, fluid connective tissue, blood contains fluid connective tissue contains plasma, RBCs, WBCs, then platelets. So RBCs, as I have told that it is by concave shape. Okay, it is integrated. It is not this, right? And it is amoeboid shape. And what is the different types of WBC? What are the different types of WBCs? Neutrophils, vesicles, cytophiles. Now this is the muscle tissue. Muscle tissue. Striations are there, nucleus are there, myofibrils are inside this filament that contains actin and myosin. Okay, and muscle fibers. These are myofibrils. And these muscle fibers can contract and relax. Contraction means shortening and relaxation. Like if you can do like this, there's a contraction and relaxation. So, along with the skeletal tissue, muscle tissue helps in locomotion and movement. It also helps in locomotion and movement. Next, there are three types of muscles: skeletal, smooth, and cardiac muscles. Skeletal means it is also considered known as striated because you see here striations are there. These are voluntary muscles. Voluntary, why voluntary muscles? Why voluntary muscles? Then muscles move according to our will. If I'm writing on the board, this is voluntary muscle. And smooth muscles, as the name indicates, there will be no striations, right? And the cardiac muscle, as the name indicates, it is present in heart. Now, first is the skeletal muscle. You see here, skeletal muscles. These are attached to the bones. It is forming biceps and triceps, right? And striations, see your striations are there. These are light and dark bands. Striations are there, and these are arranged parallelly. And it has a nucleus in it. So a sheet of this tough connective tissue encloses several bundles of muscle fibers here. Biceps, and this figure is showing biceps and triceps. And next is smooth muscle fibers. These are the internal organs, right? It is spindle shape and is involuntary. And in alimentary canal, these are present, smooth muscles are present because there is a peristaltic movement, right? So no striations are there, just present in the wall of internal organs. Apart from that, blood vessels, stomach, and intestine. These are present in this small intestine. It has big vessels, spindle. And this is cardiac muscles. And these are involuntary muscles present in the heart, and these are branched. Intercalated is called intercalated disc, right? And it also has a nucleus. So it is involuntary in nature. So only one muscle fiber is voluntary in nature. Rest of the two are involuntary. Which one is voluntary in nature? Skeletal. That is why we can able to move whenever you want to. Is it clear up to this? The next is nervous tissue, neurons. It's a neuron. This is finger-like structure is dendrites. And the red part is cytosol. And this yellow one is nucleus. This blue one. And the red part in between the two one, rhodopharyngeals. And this is axon terminal. The other neuron is attached here. What happens now? This dendrite, it just picks up the impulses. And from here, that electrical impulses enters to cytosol to axon, then to axon terminal. And see this globulated part. This is synaptic cleft. Means there is a gap between the two neurons, and that is called synapse. Information is transmitted from one neuron to another through a neurotransmitter. And what is the name of that neurotransmitter? Name neurotransmitter. Neurotransmitter. Synapse is the gap between the. You are feeling silly? Synapse is a gap between the two neurons. All these things we have already studied between the two neurons. And neurotransmitter, it just transmits the signal from one neuron to another, and the neurotransmitter is acetylcholine. Acetylcholine. This is the neurotransmitter. A synaptic cleft here, this part, lobulated part, it gets activated as the signal passes through it and just transmits the signal from one neuron to another. And there is a gap between the two neurons at this synapse. This neuron you will study in nervous system. It helps in control and coordination. We have not studied no chemical coordination in class. Control and coordination. Right? That is nervous system only you will study in this. Control and coordination is CPSC, that is in this nervous system you will study this neuron acetylcholine. It helps in the control and coordination of the body. Because it is just forms these nerves, they are interconnected with each other. Nerves are not a single chiplet-like structure. No? Various neurons are attached with each other, connected with each other. So that it is the network of neurons. Is it clear? What type of connective tissue is characterized by having a gel-like matrix and is commonly found in intervertebral disc? Which type of tissues are present? Connective tissue is present in a gel-like matrix. Hyaline cartilage. Good. Then there are two cartilage now. So bone and cartilage. Which of the following statements regarding apical meristem is incorrect? It brings about increase in length of the root and stem. It dwells by dedifferentiation of permanent tissues. It constitutes primary meristem. None of them. Very. The walls of Koch cells are thickened by the deposition of an organic substance which makes these cells impermeable to water and gas. This substance is suberin, Koch cells. Lignin is present in xylem. Suberin is present in Koch cells and this pectin is present in cell wall. Cell wall. What is inside? It's a compound. Lipid and protein. Cell membrane is made up of lipid and protein, and cell wall is made up of cell. Okay. Then, which of the following epithelial tissues lines the stomach of intestine? This one, intestine. Out of these three. Third one, this second one. Intestine and stomach. Absorption and secretion of. So it is columnar. This is cilia. It is present in respiratory tract. Okay. And these are columnar skin cells, right? Now, in the achiever section, you will get such type of questions. Refer to the given figure. Is it visible to you? Or you can sit there. Second one is A. Okay. Okay, proper position, you can stand here also. Side face and you can stand here also, no issue. What about the first one? The localization within the cells. A, B, C, or A or D. What is A? A is the set. See here, you can see endoplasmic reticulum. Your endoplasmic reticulum. So what it can be? Yes, nucleus. So chromosome is in the air only, no? This means of sequestering many of the mRNA. mRNA is in the C. Not that is RNA. A. Then next, second. Set okay. From there, you can see this. T. A. Is out there. This one, no? There's the osmosis. The two fresh potato PQR were immersed in a solution concentration. What about the second? This is A here. This is A. Actually, you are looking for this. This A. Answer is A only. Yes. A only. This all the three are for A only. What is B? Yeah. Is the nucleus. Very good. And C. Nucleus. And D. Section part of this nucleus. That was all A for this chromosome ka kinetochore. And this 4 strips of fresh potato PQRS were immersed by very good. So let us recall all the slides. I've taken only 2 questions on the or if you want, we can discuss those questions also. Let us discuss those. Mother, you know? This is caused by single and triple cylinder cell membrane. I think there at table. See, it has the lipid binaries. These are binary, hydrophobic end and hydrophilic end. These are attached like this. Okay, and the proteins are present. 3 proteins are present on the top of this layer or in the middle, or the middle. It just helps in passing on the solutes or ions from outside to inside. It is present in cell membrane. Okay. So it can be, the answer will be lipid bilayer something else. which iron holds ribosomal units together, that is magnesium ions. Thus, pigment molecules of chloroplasts are located in the walls of the okay. Halogens are present in the chloroplast. Term chromosome was coined by Goldberg. Cyclopean. Then the phospholipid form of fibromolar here in the middle part. Answer I gave it now. Have you gone through these? Vulgarities are present plant discollaging chromosomes. Next, pegosomes and pitosomes. I didn't tell now. Tell. So, this vesicle that engulfs, right? Right. And pitosomes by pinocytosis. Strength and rigidity of cell wall is due to cell wall. The strength and rigidity. Remember this word? The plant cell. Lignin that gives strength and strength. Very good. Let's use the description of ribosomal units. Stands for I explained regarding ribosomes. Student is there. Ribosomes, it has two subunits, 70S and 80S. It is in prokaryotes and it is in eukaryotes. Right? This 70S, this S is what? This S is Svedberg's unit. You know why it is called Svedberg unit? Because because of the name of the scientist, that is Theodor Svedberg. And measures the sedimentation coefficient. Sedimentation coefficient. And this we have not done the process now. So if it is there in minuses, then where it's separated. Anaphase. Right. That's true. So anaphase. Okay. This one. Anaphase 1 means it just starts. Anaphase 2 start. Next. Age of tree. Equal arms. Pencil. size. Small muscle fibers. Seriation plants the skin. Seriaceous gland. This seriaceous gland is called zebra. Armstrong is Armstrong. What number? Because why no why? Because each colonial cell is four colon mother cells. It's four, so it is this is asparagus. There is no mistake. That was it. Storage. Macromolecules. Peroxide cycle. Or we are continuing again. Phospholipids. Lipid molecules. This we have done. And then gone through this. Yes. We have discussed regarding bacteria, their role. Then this is glandular border. Ribosomes we have done polyphenotic and alcoholic membrane. The side model of the same qualitative chromosomes. Cluster of chromosomes. Okay. That contains thousands of DNA strands. And large chromosome this qualitative chromosomes is Bagga. So the development of this baby. We have done silver we have done lysosomes, antibodies, keratinocytes, bacteria. You want to see the slides because of these things? We have got them. prokaryotic cell, this is we've understood this. This one, like lysosomes, chromatopause, diploid in the bacterial cell. Chromatopause is present in cyanobacterial and these are the important in some prokaryotes. Function is same like that of pigment containing membranous proteins. That's the thing on this one. Yes, that's this ribosomes are on this one. Ribosomes. The present in some cells. Cells and tissues are common. It's chapter take time. Why mitochondria is considered as a powerhouse? Because it helps the respiration of the cell and synthesis of ATP. Liposomes, helpful then this year understood by the colleges and mitochondria. This is plastics within plants only. Ribosomes. Regarding chromosomes, we have structures. Cell division. That tissues. What? This is again mixed. You must have seen, observed that if you cut the tip of the plant, it will start growing sideways. Not stop, it will stop growing from tip now. Because of the apical meristem only. This chlorophylline. This is lignin chlorophylline. Oxals, stomata. This is bidirectional. Why that bidirectional movement occur? Because it needs to go within those parts and also from where? From the leaves. Because it manufacturing. So it has to go upside down. That is why if you water the plant or soil or the soil cut-ups. Which part of the xylem cut-ups? Water? The xylem tracheates and the xylem vessels. Very good. And what is the function of xylem parenchyma? Stores. And here in front, it develops food control. Why? They have a corporation there. Animal tissues. Tissues. This tissue is there in the 9th standard. I think all everything out. Next part of this. Actually, this exams follow NCR scripts. And since you are nice, you will be much benefited. This is from this you are studying X-ray system. This is the structure function unit of. And what is the function? What is this? That is the experiments. Let's still read. Look at this. Ultrafiltration occurs here. Micro, this multi-digit. Still started it. And villi, what is the role of villi? Does it cause intestine? It has absorption because it has the enzyme. That's the thing. And the function of this, what is the purpose of this villi? It surface area of absorption. Cilia, ciliated pulmonary cells are present in the respiratory tract. Tubes, bronchioles means it just removes the mucus from the respiratory tract. And goblet cells, all the connective tissues you have understood. Go down with this. Okay. Stop sharing. Yes, Jaishnavi. Have you understood? Actually, I won't be. You are not. I'm not able to hear you from the very beginning. That is the problem I was facing. If you want to ask anything, you can just ask me. There is a technical issue. You have understood everything. All the topics you have understood. If you want to see any of the slide, if you want to understand, you can ask. Just type it. Very good. So we'll meet on 16 with the next chapter. Okay, we'll meet on 16, and I want you to just go through the booklet. Okay, go through this booklet, so it will be easy for you to understand the next topic. What is the next chapter? Nutrition and yes, nutrition. Nutrition. Okay, Jaishnavi. So we'll meet on 23rd, 16, like 16th. We'll meet on 16th because 9th will be the 10th will be the holiday, no? 9th, Rakhi. So we'll meet on 10th. So you just read the booklet which you have given. Okay, nutrition and excretion will go with that chapter. Okay, and bring maximum of doubts. Okay. And next time I will make more questions. I will try to take more questions. That will be crazy. Jashnini, you have done all the questions I have asked that. Okay, good. So see you on 16. Bye. Still we have time. You can ask. You have 10 minutes. You can ask them. You understand what is it? It just helps in the exchange of ions from protein. The integral proteins are there. Just see. Lipid bilayer molecule. No? Protein molecules are present here. It has hydrophilic end and hydrophobic end, like this. Hydrophilic and hydrophobic, I think you must be knowing what is hydrophobic. So funny. Yes, same thing. And what happens that this model, next time I will explain that model. The protein molecules are present here and there. So in between this, it is embedded. Okay, and then it has an exchange of ions. Signal and the nickel for this also. Okay, any other topic? All everything is clear? Okay. Jaishnavi, any of the topic you want to ask? All is done with you? Clear everything? Glandular epithelium. Yes. See, the upper layer of the epithelium, when there is enfolding, it forms goblet cell. This is called goblet cell. Okay? And this enfolding, it just increases the surface area, right? So, where it is present, glandular epithelium. Yes, Vaishnavi? Yes. Yes, goblet cells, Vaishnavi. Which cells these are present? Sweat glands. Salivary glands. Goblet cells, yes. Have you understood this? It increases the surface area, like that enfoldings. Right? This is called glandular epithelium. Because it is the extension of epithelium. Right? Anything else? Tissues? Animal tissues? Plant tissues? Everything is clear? Cell? Okay. Okay, bye. Take care. Can leave the class. Bye. Take care.